Hello everyone, my name is An Ouyang. I'm the product manager of Apro Biosystems. Today it's a great pleasure for me to present our recent work and recent developed products, and it's about the selection of the AAV capsids by evaluating transgene delivery using the human organoid models. So we know there's a huge gap between the preclinical animal studies and the human studies. And the major reasons is because of the genomic things. And also in the preclinical studies, they're just using the single cell type. And that is totally different to the real world. And the human tissues, that's involved many different cell types. And the organoids is the bridge to fill the gaps and to solve the pain points, and can also to provide the high translational rate from preclinical animal study to the human studies. And our mission is to provide or offer the solution tools to the industry, to the human communities, and to solve the pain points, and to support and accelerate the drug development and the drug safety evaluations. And here I'm very proud to introduce our recently developed the organoid differentiation kit. And here I will use two of them, the cerebral organoid differentiation kit and the cardiac organoid differentiation kit. Besides these two, we also have a liver and intestine organoid differentiation kit. Let's look at the differentiation results in the phase 1. Following our internal validated protocols and torture the cell lines derived from humans, iPS, and after the torture and the differentiation, you will see the single cell become the organoids light and have the different cell types. And you can see the size and the edge of the shape. And I want to highlight at the day 4, there's the EB formation. That's the key timing to transformation from 2D to the 3Ds. And let's look at the different cell types in more details within the cerebral organoids. First, at the early stage, we use the immature, the neuron staining, and that is to tell the neuron progenitors. And here we stand with the nesting and the TUJ1, that's the beta tubulin 3. And you can see the green dots and the blue dots. That shows that's the early stage. There's the different neuron progenitors, and with the times going on, you will see the mature neurons, and here the first is the dopamine-genic neurons, and we stand with the MAP2, and also the tyrosine hydrolases, and these are the biomarkers to show the dopaminergic neurons. And additionally, we use this to show the mature neurons of astrocytes, and here we stand with the GFAP. That is the biomarker of astrocytes. Astrocyte is the very important neuron cell types, plays the pivotal functions of the brain. And then let's look at the phase E, and besides the astrocells, and we have to show that oligodendrocytes with the staining with the OLIG2, and to show that the specific cell types. Additionally, for the dopamine-genic neurons, we stand with the tyrosine hydrolases to show that this is the microglial. And so overall, the phase 1 listed here to show the progress of from the human-derived iPS from just the cells to the cerebral organoids. And these organoids finally involved with the many different cell types, and to show that this is a real brain. So this is the kind very different from just the single cell lines. And additionally, besides the morphology data and the histology data, and we have do the characterizations of the cerebral organoids, and we do the transcriptomics and to verify the different transcriptome factors, and they did have the overexpress, and that indicate these proteins is expressed at the functional level. Additionally, we do the electrophysiology testing. That's very important for the cerebral organoids. That means these different cells, they can play their own functions. And here I want to show you this data. These they have the different probes or channels that traverse the whole cerebral organoids. And these probes can receive the electrical response from the organoids and generate the feathers listed here. This is very similar to the real clinical diagnostics using the EEG feathers. You can see the pits upside down from the different channels and different response waves. So combined with the electrophysiological testing and the histology testing, that means this is the mimic of the human brains. So that's the huge advancement than using the single cells or comparing using the animal model, because this is a human iPS derived. So that's the mimic of the human real brains. And additionally, this is the normal or the healthy organoids. And based on this, we can build up the disease model of the Parkinson's disease. And here we use the alpha-synuclein preformed fibers. And it controls the aggregation of the fibers. And with the staining here, you can clearly see the difference. Here, with the time going on, you can see the fibrous network structures that is considering right now as the root cause of the Parkinson's disease. So this is not just only the healthy, the organoid models, but to be also used for the disease build-up, and also to allow the researchers to evaluate the mechanisms of the disease. And additionally, we have the disease model, and then we want to develop the drugs to combat the disease. And here, the gene therapy is becoming the most popular, most popular therapeutic strategies. And here we have the AAV screening. First, you want to use the AAV as the vector to deliver the genes to the targeted regions. But first, you want to evaluate the delivery efficiency. Here, we have the 3 different types of the isotypes of the AAVs. First is the AAV wild type, AAV5, and the IVB1 and IVB2. They are 2 different modified by our partners. And the gene incorporates with the EGFP and to show the markers. And you can clearly see that the difference. The IVB2 has the highest delivery efficiency to deliver the target, the therapeutic genes to the cerebral organoids. So with all the 3 data from the healthy organoids build up, and the functional data testing and disease model and the drug screening, this is the total solution or comprehensive solutions to support the neuron disease, the mechanism research, and the drug development. And additionally, we have the cardiac organoids. And similar to the cerebral organoids, this is the tortuous steps. And here I want to highlight the figure 4C here. This is actually a video that can show these cardiac organoids are really beeping. So it's the same as the heart. And also with the additional staining of the biomarkers. And here we are using the MLC2A or MLC2V. That is the 2 biomarkers for the atrial and the ventricular cell types. And you can clearly see the 4 different chambers, and at the top panels, top level, that is the atrials of the heart. And at the bottom, that's the ventricle of the heart. And so this from the beeping, the video, and the histology, the images that shows this is clearly the mimic of the human heart. And we know the heart is not just the chambers, but also includes a lot of the vasculars around the heart, like the coronary artery. And we use the CD31 as this is the endothelial cell, vascular endothelial biomarker to stain the cardiac organoids. You can see that the red dots covers all parts of the cardiac organoids. So this shows that it's not just a single heart, but also covered by the heart-related blood vessels. And here we do additional the electrophysiological testing. And here, compared with the other models, our model can produce the consistent and stable heartbeats. The heartbeat is at a very stable pace. And also the beat amplitude is very stable. So you don't want your heart sometimes beat fast or sometimes beat slow. And our model is the best one here. And additionally, we have the silicon probe recordings with chip arrays and to show the different size of the cardiac organoids tend to have the different response. And here we have the MEA analysis. And this is the cover the hearts and receive the response, electrical response from the heart from different channels, ion channels. And here you can see the feeder. This is very, very likely as the human ECG used in the clinical diagnostics. And so this data is the best proof that this is the real heart and the heart has its own function. Additionally, we know that the drug toxicity and to evaluate the drug safety need to cover the neuron toxicity, cardiac toxicity, and the liver toxicity. Here, we used our cardiac organoids to do that, and we have introduced the drug A to the cardiac organoids. The up panel, that is the control, and the bottom panel, that's the introduced drug A. And you can clearly see the difference for the vascular and endothelial cell biomarker CD31 and also the cardiac troponin. These you can see with the introduced drugs. You can clearly see the intensity of the fluorescent. They are significantly lower than the control panels. So that means these drugs at a specific dose, they are harmful to the human hearts. And also these could be used to do the drug screening. And previously I mentioned it could be used to select the AAV capsids for the neuron disease. And here we do the similar work. We found that IVB2 was the more optimal the gene delivery vector to deliver the target genes into the heart, and which is the result we found in the cerebral organoid. And so this gives the ideas when the drug developer researcher, and when you want to employ the similar or the same the gene delivery vector and deliver the target genes into different regions, and they can apply our cerebral organoid and the cardiac organoid to do such work. To find out the best AAV capsids, and our kit is the 96 well plates. So it allows you to do the high throughput in a short time, and this is the well images showing here. So finally, I want to conclude that the cerebral organoid differentiation kit and the cardiac organoid differentiation kit, they are the powerful tools to support the community, both in industry and academia, to accelerate the drug research and development at the early stage to understand the mechanisms of the disease and to be used to screen the drug candidates and to be used as the very powerful tool to evaluate the safety and the toxicity of the drugs. And we are welcome the partnership within the industries and also the academia. And if you want to learn more about our powerful tools and also our other products to support the drug development, please visit the aprobiosystems.com for more information.